Hello everyone. In our line by line NCRT biology explanation, in the last video we went through generally about bryophytes, and now we're gonna get into details about the two specific types of bryophytes, which are liverworts and mosses. Okay, so without bitching, so after practicing this, we will go and practice in Mnemonite the full bryophytes. Okay, so liverworts. So liverworts grow usually in moist, shady habitats such as banks of streams, marshy ground, damp soil, bark of trees, and deep in the woods. Okay, so this is just a highlighting. Like all the gymnosperms live in moist, shady habitats. So additional information given is they are live in bank of streams, marshy ground, damp soil, bark of trees, etc. All these areas are moist areas. Okay. The plant body of a liverwort is thalloid. It's not a thallus, it's a thalloid. Thalloid in the sense it is still not differentiated into root stems or leaves. Because what do you mean by thallus? Thallus is a plant body that is not differentiated into root stems and leaves. But a thalloid is something which is still not differentiated into root stems and leaves, but has some structures that are differentiated into root stems and leaves. Like, I mean, intermediate. It is not completely differentiated, but it might look as if it is differentiated. So we will talk about that soon. So example, Marchantia. So this is a type of plant, type of liverwort. Okay. So the thallus is dorsiventral. So what is the meaning of dorsiventral? Okay. So dorsiventral is nothing but if you take a leaf, you will see that one side of the leaf is dark green, and if you turn this leaf and see, the other part of the leaf will be light green. So dorsiventral, in the sense the thallus is different on both the sides. In the dorsal side it is different, and in the ventral side it appears differently. Okay, I can even show an example of a dorsiventral leaf. So there you see, this is a dorsiventral leaf. You can see that the front side, dorsal side, is dark green, and this is the ventral side. So it looks light green with a lot of small branch like structures, right? So this is what is called as a dorsiventral leaf. Okay. So see, the thallus is dorsiventral. So in liverworts, the entire plant body is dorsiventral, right? Like it looks different in the dorsal side and the ventral side. It is closely oppressed to the substrate. Yeah, that's true. It is. It is mostly like it is. It is like it is always oppressed. In a sense, it is, how do I say, it does not grow very tall from the substrate, right? It is very in nearly close contact with the substrate. So that's what it means. So the leafy members have tiny leaf-like appendages in two rows on the stem-like structures. Okay, so I can definitely show you this in another image. Give me a second, because it is very important. Show some images of liverworts. So you see, it is not of single layer. Where do I show you? So if you see here, see, so this is the layer below, and there's one layer of leaf which is above, which is still leaf, right? So that's what they're telling here. They're telling that the leafy members have tiny leaf-like appendages in two rows on the stem-like structures. Okay, so stem-like structure. So there is the meaning. Like you see, uh, say for example, like which is a better image I could show, is probably this. I don't know. I guess it's understandable, right? Like layers above layers. Okay, so that's the point here, basic point. So that's what they're saying. They're saying that the leafy members have. So suppose this is the leaf. It has other leaves, small leaf-like structures. Okay, with a small stem-like structure here. So this is how the plant body looks like. So this is a large leaf, and above this it is small leaf-like appendages on stem-like structure. So that's how it grows. Okay. So leaf-like appendages in two rows and stem-like structures. So asexual reproduction in liverworts takes place by fragmentation of thalli. So so this is the thallus. Like if it just breaks, like fragments, or by formation of specialized structures called as gemme. This is a key word. So asexual reproduction in liverworts is through gemme. So gemme as the plural of gemma. So gemme are green multicellular asexual buds which develop in small receptacles. So receptacles are cup-like structures. So it's simple. They're again telling gemma cups. Okay, so receptacles are just cup-like structures. That's it. Gemma cups located on the thalli. So this is shown in this diagram. Not in this diagram. I will show it here. See here. Gemme, the cup-like structures in this image. See this. So these cup-like structures are called as receptacles, but for liverworts it is called as gemme. Okay, so these cup-like structures. So these cup-like structures originate from the thalli, and they can also help the liverworts reproduce asexually. Okay, and then the gemme become detached from the plant body and germinate to form new individuals. So that bud-like structure. So that a bud-like structure. So this actually like breaks from the plant, and again it germinates to form a new plant. Okay, so that's how the gemme works. And during sexual reproduction, the male and female sex organs are produced either on the same or different thalli. Okay, so the sporophyte is differentiated into foot, seta, and capsule. Okay, the sporophyte that comes out of liverworts, so they are divided into foot, seta, and capsule. After meiosis, spores are produced within the capsule. These spores germinate to form free living gametophytes. So they're just telling the male and female sex organs are produced either on the same or different thalli. So one thalli can have both the antheridium and archegonium, or it could be antheridium in one plant and archegonium in a different plant. So it could be like that. And then we know how the sporophyte forms, right? It has been made clear here. The sporophyte forms from a zygote. So that sporophyte is differentiated into foot, seta, and a capsule. If I have to show you the image, I can show you an image like how it is divided into foot, seta, and capsule. So look at this sporophyte. So it's a foot, seta, and in the top there is a capsule. So this is how a sporophyte looks. So this is a moss picture, but still it is applicable for liverworts as well. Okay, the sporophyte that comes out of the zygote is differentiated into foot, seta, and the capsule. And why are they talking about capsule here is that after meiosis, the spores are produced within the capsule. Okay, so in this process, we've seen like generally in gymnosperms, what happens? The sporophyte then produces, like undergoes meiosis and produce haploid spores. That spores will become gametophytes. That's the same thing here. So after meiosis, the spores are produced within the capsule. By meiosis, in the capsule. Like, they're just telling this sporophyte has a capsule, and from that capsule, meiosis happens, and the spores are haploid spores are produced, and these spores germinate to form free living gametophytes. It is just the basic thing. If you understand how generally the bryophytes reproduce, this is just some details are added. That's it. But the process is the same, right? The sporophyte capsule and the spores germinate to form free living gametophytes. That's it. The process is simple. So then when we have to talk about mosses, so this